Thank you and welcome to join our online webinar series. As Miranda introduced, we have topic cover a lot of applications, starting from transcriptomics, genomics to epigenetics, which will be today's topic. Myself, my name is Yuanyuan Chen. I am currently the sales director for Novogene. This year will be my 10th year working at Novogene. So we had, I witnessed some really great achievement and exciting development regarding our services. So we're happy to dive into that today and then give you a bit overview what can we help you with on your research. Before we start the seminar on the technical part, please give me about 5 minutes to have a short introduction of Novogene as a company. So Novogene is a global company. Right now, we are running 10 global laboratories across the globe. And we have 19 subsidiaries around the world. Our business covers around 90 countries and across 6 continents. For those of you who are working in Europe, so these 2 European labs are where your samples will be shipped and processed. We are running the Cambridge lab in the UK ever since 2016 or 2018. And we recently opened up a Munich laboratory starting beginning of last year, 2024. And this lab has been running 1 year and a half now, quite smoothly. So if you're based in Germany, for example, or mainland Europe, this will be your top choice regarding the sequencing lab. Novogene can support a wide variety of applications, ranging from basic research, populational genetics, complex disease, to infectious disease, and productive health, a lot of different applications. For our services, we really covers a very comprehensive portfolio. No matter what research area you're in, I'm sure we will help you find appropriate solutions. Just want to mention that we recently this year launched 2 exciting new services, including the spatial transcriptomics and single-cell RNA-seq supported by the 10x genomics technology. Also, we this year launched the Olink service for the proteomics. So if your research involved or plan to do these, please also get in touch to us. Today's main topic, we will cover the epigenomic part. We will dive into what are the technical backgrounds and what are the available services that Novogene could provide. Why Novogene? So we listed a couple of reasons, including reliable results, large capacity, latest technology, fast turnaround, expert analysis. I'm sure that you will find Novogene as your trusted sequencing provider, and especially we are rooted and planned to serve in the long run in Europe. So based on the 10 years of operation experience, we will keep expanding our portfolio and bring better, faster, and more user-friendly services to all the researchers. We are always running the latest sequencing platform. We are running now Illumina X Plus in both UK and Germany lab. And we also have PacBio locally in Munich lab. We also run the nanopore platform to serve different needs of your research projects. We keep publishing high-end journals. and developing our own in-house software and pipelines, and then supporting front-edge scientific discoveries. You will find our sales team within majority of the European countries, and you will get contacted by your local representative who will live about 2 to 3 hours away from you within the radiation, and we speak your languages, and we are ensures that we help you reach your scientific goal. And with that, please let me to introduce today's speaker, Aman. He is our product marketing manager. Today's topic is gonna be epigenetic evaluated NGS solutions for transformative epigenetic research, and looking forward for that. And Aman, here's your stage.
Thank you so much. Thank you so much, Yuan, for the kind introduction. I'll start sharing my screen. All right, I hope my screen is visible. Perfect. All right, so let's begin. So today's webinar is going to be about epigenetics sequencing, and I'm going to discuss some epigenetic solutions that Novogene can provide. So I will start with methylation sequencing, and we discuss different solutions that Novogene can offer you for methylation studies. This is going to be followed by discussing ChIP-seq, RIP-seq, ATAC-seq, and a little bit about non-coding RNAs. So starting with the definition of epigenetics. So epigenetics is essentially the study of how organisms control gene activity without changing the DNA sequence. So these epigenetic mechanisms, they allow organisms to respond to their environment by altering their phenotype, but without a change in genotype. So there is no mutation as such. Now, these epigenetic modifications can be of many different kinds, and some of these epigenetic modifications are also heritable, and some can also be reversed. Epigenetics plays a key role in development, in aging, in health, disease, and several other processes. I also want to mention the term epigenomics, which is the study of the complete set of epigenetic modifications across the genome. Now, as you can see in this image here, epigenetic modifications are of many different types. And we need tailored sequencing solutions to study each one of them. So we will start with methylation sequencing, and starting with the definition. So DNA methylation is the biological process by which methyl groups are added to the DNA molecule. Now I want to highlight here that so far it's adenine and cytosine methylation that has been studied well. And in eukaryotes, we see that cytosine methylation is the most widespread. Also in mammals, DNA methylation predominantly occurs in CpG dinucleotides, and it can be found in less frequently in other contexts. And we see that for other kinds of organisms like plants, we see a lot of methylation actually happens in these different contexts. Now, as far as the function is concerned, when located in a gene promoter, it typically represses gene transcription. And when it's located within the gene body, the function is not very clear. So why should we study DNA methylation? So studying DNA methylation provides us with a lot of information that can be used in clinical research, for example, in cancer research, or in studying mechanisms of different kinds of diseases. It can be used in plant and animal research, for example, in the area of agriculture. It can be used in developmental biology research, for example, when we are trying to study the mechanisms of cell differentiation or tissue development. And it's really powerful because just like RNA-seq, you can identify some differentially methylated sites. So just like RNA-seq, where we have like differential gene expression, and you can compare different kinds of conditions or samples or groups. Similarly here with DNA methylation, you can do that and you can really make some significant conclusions. So there are different kinds of sequencing platforms also that can help you to detect DNA methylation. So we use Illumina, PacBio, and Oxford Nanopore. PacBio and Oxford Nanopore allow direct detection of DNA methylation. This is something that I will also mention briefly, but largely we will focus on Illumina-based solutions. So starting with whole genome bisulfite sequencing, this is considered to be a gold standard of methylation sequencing, and it measures methylation of single cytosines. So how does this technique work? So we treat our DNA with sodium bisulfite, which is a chemical, and this converts unmethylated cytosines to uracil, while methylated cytosines, they are not converted. They are protected from conversion. And finally, when we do the PCR and when we prepare the libraries, these uracils change to thymine. And after sequencing, you can compare the data with the reference genome, and you can try to infer which sites were methylated. Now, the advantage of this technique is that, yes, it is for the whole genome, and it can identify methylation solution. So there are different ways of preparing libraries for bisulfite sequencing. Now, if we look at the image on the left here, this is like the traditional method or the method that was followed earlier. Here, we first carried out bisulfite conversion, and then we did the library preparation. Now, the problem with this method was that we observed that there were a large number of broken library molecules. And in order to get sufficient quantity as well as quality of data, we required much more amount of samples. So the sample requirements were quite high. So there has been an improvement and advancement. And we have another method, which is called PBAT, or post-bisulfite adapter tagging. Now, in this method, we first do bisulfite conversion, and then we do the library preparation. Now, we have observed here that the libraries are of much better quality, and we need much less amount of sample. Also, I want to mention that at Novogene, we also run a control to check the bisulfite conversion rate, and we report that to you so that you have confidence in the technique that we have performed. So just to briefly summarize, whole genome bisulfite sequencing is the gold standard for methylation sequencing. It can identify methylation in all different contexts at single base resolution. At Novogene, we provide full service, so we can also offer you bioinformatics analysis. Also, I want to mention that the sequencing strategy is going to be paired N150. Our recommended data depth or amount would be like 30x or more. And the sample requirements can be accessed online. Now, moving on to another technique, which is reduced representation bisulfite sequencing, or RRBS. This is like a subset of whole genome bisulfite sequencing. And here we are enriching for the CpG-rich fraction. So how we do that is that we use an enzyme called MSP1, which sort of attacks on those particular CpG sites. And we are able to enrich for those locations. And then we can do the bisulfite library preparation. So this method allows us to study methylation just in the CpG context, which means that it is a great way to study methylation in mammalian systems, where we have methylation primarily in CpG context, and it gives you a smaller, manageable data set. Finally, another technique came up a few years ago, which is enzymatic methyl sequencing. Now, this technique relies on the use of specific enzymes to distinguish between methylated and unmethylated cytosine residues in the DNA sequence. So it primarily utilizes two enzymatic steps, and the reaction conditions are much milder. So if I compare it with whole genome bisulfite sequencing, then you will observe that the overall method is very similar. So for example, in bisulfite, we see that the methylated cytosines, they are protected, while the unmethylated cytosines are converted to uracil. The same is happening here too, but we achieve it by two enzymatic steps. So in the first step, we use TET2 and oxidation enhancer, which helps to actually protect these methylated cytosines. So it sort of modifies them a little bit. And then we treat it with the enzyme called ApoBec, which actually then converts these unmethylated cytosines to uracil. And then it's the same as whole genome bisulfite sequencing. We do the PCR, we do the library preparation, followed by data analysis. So what is the advantage of this enzymatic method? So this, first of all, minimizes damage to the DNA. It's much gentler. It means we can get higher quality libraries, and it can be used for diverse sample types. We also see that it has more uniformity and reproducibility of GC coverage. And as I mentioned before, it results in the same converted sequence as whole genome bisulfite sequencing, which means that we can use the same set of bioinformatics analysis pipelines. So this is going to give you an overview of our bioinformatics pipeline. Since this is not a webinar for the BI part, so I won't go into the details of how this analysis is performed. But I want to mention that Novogene provides bioinformatics analysis, and we have data reported in the form of several figures, graphs, tables, which can also be used for publications. And we are not just performing methylation calls, but we also provide functional analysis. Now let's move to methylation detection by long-read sequencing platforms. So long-read sequencing platforms from PacBio and Oxford Nanopore technology can help us to directly detect DNA modifications. And this happens because, so for example, if we take PacBio, now in PacBio, it records the kinetics of incorporation of these nucleotides. So when there is an epigenetic modification, for example, when there's a methylation of the cytosine, then we see that there is some kind of a lag in the kinetics that is recorded. And this record can then be analyzed, and we can try to identify the methylation. Something similar happens for Oxford Nanopore technologies as well. So here we are trying to record a change in the ionic current or a characteristic drop in the ionic current when these bases are moving through the nanopore. And we observed that when there is a modified base, then there is essentially a characteristic drop in the current. Again, this data can be analyzed, and we can try to identify different epigenetic modifications. Now I'll move on to ChIP-seq and RIP-seq. So ChIP-seq and RIP-seq can be used to study DNA-protein interactions or these macromolecular complexes. And these DNA-protein or RNA-protein interactions, they are important for regulating gene expression and function. So if you're talking about studying DNA-protein interactions, we are mainly talking about here chromatin immunoprecipitation. And for studying RNA-protein interactions, I'm going to talk about RIP-seq, which is RNA immunoprecipitation. So just quickly going through ChIP-seq and the principle behind this method. So ChIP-seq actually combines NGS with chromatin immunoprecipitation to actually identify the binding sites of DNA-associated proteins. So we can identify what sequence of DNA was, for example, bound to a histone protein or a transcription factor. Also, it's important to mention here that there are several variations of this technique, for example, cut and run and cut and tag, with slightly different methodologies and applications. Now, if we just look at ChIP-seq, so how this technique works is that first of all, we cross-link the DNA and the protein complexes. We isolate the chromatin, we fragment the DNA, and then we can precipitate it with protein-specific antibodies. So in that way, we can enrich for these protein DNA complexes. We can remove the protein, and we can construct then the library followed by sequencing. RIP-seq works similarly, except that it is for RNA and can be used to map specific protein RNA complexes. And RIP-seq also has several variations. So again, because it works in a similar way, I won't repeat the details, but I want to highlight that the overall methodology is very similar to ChIP-seq. Again, for both ChIP-seq and RIP-seq, we are able to provide the bioinformatics analysis, complete with different kinds of images, graphs, tables that can be used for publications. We can also do some customized analysis. And if you have some custom analysis requirements, you can reach out to us, and we will try our best to help you out. Also, it's important to mention that apart from the usual controls, it's important to also have experimental controls, for example, like input samples that can be used to determine whether the interactions we observed are significant or caused by non-specific factors. So for example, input controls are those that have been cross-linked and sonicated, but not immunoprecipitated. So if I go back, and if you see this image in the second, for the second heading, then this gray DNA, this gray portion is going to be your input. So we really encourage that you send us this input sample so we can do the normalization, and we can account for variations in the fragmentation step of the ChIP-seq protocol. Also, this applies to RIP-seq. Now moving on to another interesting technique, which is ATAC-seq. Now, ATAC-seq is used to assess genome-wide chromatin accessibility. So the DNA inside the nucleus, it's packaged by these histone proteins. And there are portions where this packaging is extremely tight, which is the closed chromatin. And there are portions where this packaging is lighter, or open chromatin. And because the packaging is lighter in open chromatin, we might expect, for example, more transcription happening from these regions. Now, in order to study or sequence this open chromatin, we can use ATAC-seq. ATAC-seq uses a hyperactive TN5 transposase enzyme. This enzyme binds to the regions of open chromatin. It fragments these, and it also tags these with these adapters. So this process is called tagmentation. So finally, you get these fragments with adapters added, and we can then prepare the libraries, and we can sequence them. Novogene provides data analysis for ATAC-seq as well, complete again with not just information on the sequences, but also we provide functional analysis. Now, sometimes ChIP-seq and ATAC-seq can be confused together. So I have included a slide to bring out the difference between the two. So in ChIP-seq, we analyze DNA-protein interactions, and we are trying to find out the sequence of the DNA that is bound to a particular protein. And as you can observe here, in ChIP-seq, it is important to have knowledge of the protein because you need a specific antibody for the protein of interest. On the other hand, ATAC-seq is useful for sequencing or mapping the regions of open chromatin. And here, we use a TN5 transposase. So you don't have this bias from the beginning. You don't have to use like a specific antibody. So even if you don't have a specific antibody and it's not maybe a well-studied system, you can still go for ATAC-seq. And ATAC-seq can be done in both bulk as well as single cell versions. I want to mention here that for ATAC-seq, currently Novogene Europe provides sequencing for pre-made libraries, and we also provide bioinformatics analysis. Moving on, I want to highlight a published study which actually demonstrates the power of a multi-omics approach. Because all these techniques, ChIP-seq, ATAC-seq, RNA-seq, they are complementary to each other, and they can be integrated for for insights into this complex epigenetic regulation. And I'm happy to say that Novogene can actually provide joint analysis or integrated analysis. We have options to combine methylation data with RNA-seq data, and also RNA-seq data with ChIP-seq or RIP-seq data. And this really then expands the range of our analysis, and this analysis can really help you to decode complex epigenetic regulation. So if you have requirements for joint analysis, please do reach out to us as well. Now, finally, I'll move on to non-coding RNAs. So non-coding RNAs are also involved in several epigenetic processes. They are an important player in epigenetic control. So they can regulate gene expression by modulating transcriptional processes. For example, microRNAs and other kinds of RNAs can regulate gene expression. Then they're involved in imprinting. So they're involved in these parent of origin specific gene expression patterns. And they can also guide DNA methylation machinery to specific genomic regions and can potentially regulate chromatin accessibility and others. Now, in terms of sequencing of non-coding RNAs, the challenge is that non-coding RNAs are a very diverse set, and they are present in a very low percentage in mammalian cells. So we have to identify some tailored solutions to sequence these different species of non-coding RNAs. So as far as long non-coding RNAs are concerned, we provide ribosomal RNA removal and directional library. Directional library here means strand-specific library, which means that we can try to identify the original strand, DNA strand from where this transcript is coming from. Now, this is extremely crucial for epigenetics projects for sequencing non-coding RNAs, because a lot of these non-coding RNAs actually come from the antisense strand. So that is why it's important that we go for a directional and or strand-specific library. Also, if you're working with blood samples, then we also offer globin depletion. Now, while this service is named long non-coding RNA sequencing, I want to highlight that this is also going to include messenger RNA sequencing data. If you're interested in circular RNA sequencing, then we provide ribosomal RNA removal, a directional or strand-specific library, and we remove the linear RNA. For small RNA-seq, we can provide you with an 18 to 40 base pair insert small RNA library. Again, I also want to highlight that we also accept pre-made libraries. So if you want to make the libraries by yourself and you want to send it to us for sequencing, we can also do that. So while I talked about circular RNAs and small RNAs and long non-coding RNAs, we do provide analysis pipelines for all of them, but I won't have the opportunity to show all of that. So I'm just showing you like the pipeline for long non-coding RNA-seq. And as you can see here, we not just give you the information on the transcripts that are present, but we do functional analysis, differential analysis. We can also do some kind of structural analysis. And if you're sequencing multiple RNA species, then we also have several joint analysis and integrated analysis options. So here, as we know that we are sequencing both long non-coding RNA and messenger RNA, so we will have analysis for both of these RNA species. But if you're also sequencing other RNA species with us, we can try to also integrate the analysis and we can check if there is some interaction happening between these non-coding RNAs. So with this, I will come to the last part, summarizing everything. So if you come to us for a project, then we start with experimental design. So we help you out. We discuss the research question, what kind of sequencing platform would be the most appropriate, what kind of depth of sequencing are we going for, what should be the data amount. So we try to discuss all of that with you before. And then if you want to do extraction with us, let's say DNA or RNA extraction, then we can also provide that. Otherwise, you can send your samples to us, and we can then do the quality control, followed by preparing the libraries. Of course, this library construction depends on what kind of sample you've sent to us. This is going to be followed by quality control of the library, and then the sequencing. Whatever data we get from sequencing, that also undergoes data quality control. And finally, we do bioinformatics analysis. And this bioinformatics analysis data is provided to you in the form of a report, and you get all the files and images. So just to wrap up, we discussed several different solutions for epigenetics research. Like I mentioned, epigenetic modifications are of many, many different kinds. So we need to have tailored solutions for each of them. So we discussed for DNA modifications, bisulfite sequencing, enzymatic methyl sequencing, and also it's possible to directly detect some of these modifications by long-read sequencing. We talked about macromolecular interactions and how ChIP-seq and RIP-seq can be utilized to study either DNA-protein interactions or RNA-protein interactions. We also looked at non-coding RNAs and their importance for epigenetics and how could they be potentially sequenced. Finally, we also discussed ATAC-seq. However, I want to mention that Novogene has a large number of services and our portfolio is ever expanding. So if you are in requirement of other kinds of services, for example, if you want to study chromosome conformation and you want to perform Hi-C, you can reach out to us and we would be very happy to help you. So with this, I will end my talk, and now I would be very happy to take up questions.
Hello. Yeah, thank you a lot, Arman. That is really informative. As Arman speaks, our technical support team are working on the backstage to answer some of the questions already that you wrote in the Q&A session. If we have time, we will also read it out, and also for the audience to also maybe interested in the same. In the meantime, I will pick out a few open questions that Arman, if possible, you could help us to address. So one of the questions from Elisa Cavati. So is it possible to apply the enzymatic conversions to gain information about the C methylation at whole genome level? And what should be the ideal sequencing coverage? So I think this question is related to our EM-seq service.
Yes.
And C methylation, I think it meant the CpG methylation, right? If that's the case, Arman, could you please answer that?
That's right. So that's right. So yes, we can use enzymatic methyl sequencing to study cytosine methylation. And again, like whole genome bisulfite sequencing, EM-seq is also for the whole genome. And also, again, you can study methylation, cytosine methylation in all contexts. So it's not just restricted to the CpG-rich regions, but also other regions of the genome, which means it is broadly applicable. Now, in terms of how much depth you should go for, it depends on a lot of factors. Like what your research objectives are, what kind of species you're working with, and what are the guidelines in your field of research. But in general, we usually recommend 30x or more.
Yeah. And we can also support you with project design and consultations depending on what is your end research goal. So feel free to reach out to our local sales colleagues who will bring together our technical support team to discuss your specific projects. We have also a very interesting question from Adam Whips. Can the direct methylation methodology be used to determine biological or epigenetic age? Aman, can you comment on that?
Oh, that's a difficult question as well for me to answer, because I don't have experience in aging research. However, I want to mention a couple of things here. So one thing would be that these direct technologies for methylation detection, which is by Oxford Nanopore and PacBio, they're still somewhat in their initial stages. And by that, I mean that they are probably not suitable to study methylation in all contexts. So you can't get information of all the possible modifications and at all possible sites. So this is something to keep in mind. So we do know that, for example, 5MC in mammalian species can be recorded extremely well, or 6MA or 4MC in prokaryotes can be recorded really well. But we observe that in plants, for example, not all sites are covered. So this is something to bear in mind. I would still suggest that if you are really focusing on methylation detection, and if that is your prime focus, then go for techniques like whole genome bisulfite sequencing and enzymatic metal sequencing. However, if you want to do whole genome sequencing and that's your main aim, and you want some added epigenetic data, then long read sequencing platforms might be more useful. But definitely, of course, methylation is extremely important for aging, and it has been definitely correlated.
Yeah, thank you. Okay, the next question also, I think is a common question that we receive, is from Agata Abramovitz. Do you have any experience in analysis of cut and run assays performed on cfDNA from liquid biopsies? I know that we can do cut and run to some degree, but the specific type of tissue or the sample types, could you help?
Yeah, so for these kinds of projects, you can actually send us the enriched RNA, enriched DNA, for example, in cut and run. So you can actually, use any protocol you want. What we would just need is the enriched DNA fraction and the possible controls, like the input control. And then we can perform, we can do the library preparation for you, and we can also provide you with the data analysis.
Yeah,
but also important, I also want to mention here, I forgot, that again, it's key here that you first reach out to us to discuss your project in detail, because we might have to go through your protocol to assess that it is suitable for the pipeline.
Yes, that's good to hear. We already have quite some successful project experience on cut and run samples. So please do reach out, and we will give more detailed consultation. Another question from Leah Christine Meisberg. Do you provide support in, for example, finding the right proteins to be analyzed for a specific disease of interest?
So actually, we've just ventured into the area of proteomics. So we can't provide you with direct support at the moment, but I want to mention that if you're working with human samples, then we offer actually proteomic solutions from Olink, and these solutions can actually be used for screening studies because the panels that we offer, Explore, HT, and Reveal, they're actually panels with large number of proteins, and they're ideal for these initial pilot screening studies. So you can actually perform that assay on your samples, and the power of the technique is that the data you get is actually like RNA-seq again. So you can actually do differential protein expression analysis, and you can compare your sample types, and you can get a list of these candidate proteins that you can then follow up in more details.
Yeah, that's very nice to know. Next question related to bisulfite sequencing. This is from Alicio Murgia. Is it possible to customize the panel of regions to be studied by the bisulfite sequencing?
At the moment, we can't offer customization. For whole genome bisulfite sequencing, it's anyway for the whole genome. But we do, like I mentioned, we do offer RRBS, which is like focusing on a specific fraction, in this case, the CPG-rich region. And we've also seen that while RRBS is mainly good for mammalian species, but there are publications out there which show that this MSP1 method can be broadly applicable to other animal species as well. That said, while we can't provide you with a customized panel at the moment, we do accept pre-made libraries. So let's say if you want to enrich for certain fractions, you can do that from your end, and we can actually then take up the library preparation and sequencing part. So those possibilities are also there and can be explored.
Interesting. Very good. Yes, so there are some level of customizations we could discuss in detail. I do see that in the question and answer column, a few questions related to the pricing. I can shortly comment on this question, for example, from Joanna, is the cost of RRBS much lower than whole genome bisulfite sequencing? So because of reduced representative bisulfite sequencing will sequence only a fraction of the whole genome, normally around 1%. So that means the data that needs to be sequenced will be much lower. In the case of a human genome with RRBS, we normally recommend starting from 10 gigabits of data, whereas the whole genome bisulfite sequencing with the 30x recommendation will start from 90 gigabyte of data. That will result in also difference in the pricing. So RRBS in general is about 1/3 of the cost of whole genome bisulfite sequencing. Depend on how big is your genome. So just so 1/3 to 1/2 of the whole genome. So and if you have such a project, please get in contact. We will give you more realistic price estimations. Yeah. Another question also related to the cut and run from Kai-Ting Jiang. Do you usually do both ChIP-seq and cut and run if you know the researchers choose to do ChIP-seq?
I'm not sure if I get this question properly.
So normally we would not do both unless it is requested by customer, I think. So depending on what is the research goal and what particular protein or transcription factor you are looking at. But we would definitely discuss based on your research goal and then suggest the most recommended sequencing strategies. Another very interesting one, I think, is quite a representative one from Haitam Ben Amara. Is it possible to apply these NGS analysis on, for example, FFPE sections? And what about spatial epigenetics? So there's two questions within this.
Really interesting questions. So actually, a lot of these methods are actually applicable for FFPE samples. So for example, you can use whole genome bisulfite sequencing for FFPE. Actually, the best would be enzymatic methyl sequencing, but these techniques can indeed be used for samples, and not just FFPE samples, but also some other challenging sample types like circulating tumor DNA and circulating free DNA. As far as spatial epigenetics is concerned, it's an upcoming field and technique. So at the moment, we can't offer spatial epigenetics, but we've started with spatial transcriptomics. And hopefully in the future, we can also offer spatial epigenetics. But again, spatial transcriptomics also actually can give you lots of insights. So for example, on your samples, you can, do methylation studies, and you can do spatial transcriptomics, and then you can try to correlate these two data sets to get some unique insights.
Okay, good. So another question regarding the biological replicates, I think how to set up the experiment from Ayan Hader for time series samples, so with different time points. Do you recommend any special normalization or batch correction to compare from day 14 through month 24? So I think the more general question is that if we need to collect sample at different time points, do we recommend, first of all, what are the batch, let's say the biological replicates that needed? And do we need to do any normalization or batch corrections?
Yeah. So again, this question depends really on the experimental design and the project and the specific technique that we are performing. So this would require actually a thorough pre-sequencing discussion. So we will have to discuss your project in details. So yes, replicates are highly recommended. And we would recommend that for most of these techniques, you perform at least 4 replicates, though more than 4 might be encouraged for several applications. Like I said, like if we are performing ChIP-seq and RIP-seq, then it's extremely important that we have input controls for normalization. You can also include IgG controls in that scenario. For some kind of experiments, there's also like a common sample that is used for all the sequencing experiments. So if you're, let's say, sending samples in batches, then there's going to be one set of samples that are always going to be common in all those sequencing experiments. So finally, in the end, you can use that to compare and normalize. So that's also possible to do.
Yes, that I think is also quite important perspectives to before you design experiments. So thank you, Amma, for answering that. Another question from Gazam. What services can you provide for non-model organisms? Specifically regarding ChIP-seq and ATAC-seq and DNA methylation. So I know that for sure we work with a lot of different non-model organisms, so maybe even more than those model organisms. What Novogene can do is really comprehensive. We start from even at the genome assembly stage to help you having a good genome. We will also support transcriptomics with annotations and all of that. Regarding to the epigenetics section, ChIP-seq, ATAC-seq, DNA methylation, Aman, could you also comment on that?
Yeah, Yuanyuan, I completely agree with you. We actually have experience with a wide variety of species. So starting from, archaea to humans, we have experience sequencing, different kinds of samples from different kinds of species. And a lot of these epigenetics techniques are actually broadly applicable. So like I mentioned, most of these methylation techniques like enzymatic methyl sequencing or whole genome bisulfite sequencing, they can be broadly used for almost any eukaryote. Similarly for ChIP-seq and RIP-seq, though of course in ChIP-seq and RIP-seq, the main problem is to get the antibody specific to the protein of interest. So there I can understand that we might encounter issues. But again, with ATAC-seq, you don't need a specific antibody. It's TN5. So again, broadly applicable.
Yeah, that's great to know. We also we have a lot of questions from this section, and thank you everybody for being so interactive. We appreciate it. Some of the questions, due to the very high technical detail, we will come back to you after the seminar with the more technical detailed explanations and answers. There are also a few questions that had been answered in the written format already. I can also pick out a few that could be representative for the other researchers. One is from Frank Becker. Does the genomic DNA needs to have certain QC values or input values for the whole genome bisulfite sequencing? And what is the, like a sample requirement for whole genome sequencing? What we have answered is that we do have a normal input method and the low input method for you to choose from, as normally if you have around 100 nanogram of genomic DNA, this could already be sufficient for a whole genome bisulfite sequencing. So not a huge amount of DNA needed. And we also commented that this can be also used on FFPE materials. So if you isolate the genomic DNA from FFPE material, as well as cfDNA, we can apply these methods, so PBAT methods for whole genome bisulfite sequencing on those type of materials as well. And we have also quite already some very good experiences working with FFPE and cfDNA materials from human. Please get in touch. We will give you a more detailed consultation. Yeah.
I completely agree with Yuan. And again, our sample requirements are also on our website. So you can check, and you can also perform like a sample check and quality control from your end. Also at Novogene, we do a quality control, and we also then discuss the results of quality control with you. And you can just proceed with samples that you feel are good quality, or if you want to, during the quality control, if let's say you feel that you want to maybe send another sample instead of something, you can also do that. So that is also possible. And yes, we do have experience with low input methods as well. And now, thanks to the PBAT protocol, whole genome bisulfite sequencing also does not require so much DNA. If you want to work with FFPE samples, for example, yes, you can use it. Whole genome bisulfite sequencing can be used. However, especially for these challenging sample types like FFPE, circulating free DNA, circulating tumor DNA, I would really suggest enzymatic methyl sequencing because it is really gentle and the sample requirements are much lower. So even if you have 15 nanograms or so, we can actually perform the technique.
15 nanograms. That's
Yeah.
very impressive. We also work with
impressive.
like 10 nanograms on some cases. That's I think it helps a lot of researchers to be more easy on the sample part. Yeah. Thanks, Aman. Maybe as the time is running up, I will also comment on one of the questions regarding prices. And this is from Julene. Could you provide us with an average cost per sample for the different techniques that were discussed? This is a really good question, and I know everybody is interested in this topic. For the bisulfite sequencing, epigenetic sequencing, this is genome size based. So the cost will relate it to how big is the genome you're studying, whether it's a human or this is a plant. Because of the genome size difference, the price will be different. But in general, whole genome bisulfite sequencing and enzymatic-based whole genome sequencing have pretty much similar cost, whereas the RRBS-based, so reduced representative-based, have a lower cost, so 1/3 to 1/2 of the cost. In terms of the others, for example, ChIP-seq, RIP-seq, this kind of applications, the cost is relatively similar at a similar level at transcriptomic sequencing. These days, these kind of assets had become quite affordable. So around, again, depending on what is your species of interest, could range from 100 to 300 euro per sample. So it's not a huge budget you need to plan. So please get in contact with your local account manager. We will help you plan the budget and plan the whole project out. Yeah. So I think that with the time limits, we will end the question session now. And thanks everybody again for joining us today. On the screen, you will find 2 upcoming seminars that we already have on the registration is out. So if you're interested, please also sign up to learn about these different topics. At the same time, welcome to get in touch via LinkedIn and Novogene website to keep updated with our newest service. Today, after the seminar, we will send you a recording of today's talk, including the slides, so you can anytime go back to specific details and to check on your projects. And after this seminar, as a part of the benefit, you will also receive email from Novogene, including a special discount for your next projects. This will be 15% discount. So you can apply that for your next sequencing project with Novogene. In the meantime, if you would like to have a copy of a certificate of attendance, this can be requested. We will help you print out one certificate upon request. And at the same time, thank you a lot for joining us today, and we welcome and looking forward to working with you in the near future. Thank you.